The main theme of this project is observation of active pharmaceutical ingredients by HPLC and GC. HPLC and GC is a techniques used for analysis of pharmaceutical compounds. High pressure liquid chromatography is used for analysis of non-volatile compounds, whereas gas chromatography is used for analysis of volatile compounds. Mainly HPLC is used for analysis of impurities of non-volatile compounds per percentage of purity. The GC is used of the analysis of related substance of volatile compounds. The main aim of this project is to understand principle and instrumentation and operation of HPLC and GC performing by related substance of S-naphin malate. Introduction. HPLC. HPLC is a form of liquid chromatography used to separate compounds that are dissolved in a solution. HPLC is an analytical chromatographic technique that is useful for separating of molecules that are dissolved in a solvent. If the sample solution is in contact with a second ion solids or liquid phase, the different solutes will interact with the other phases to differing degrees due to differences in absorption, ion exchange, partitionating or size. These differences allows the mixture's components to be separated from each other by using these differences to determine the transmitter of the solutes throughout the column. HPLC is a liquid chromatography. The main principle of separation is absorption chromatography. In this absorption chromatography, solute particles moves along with the surface of the stationary phase. Various physical and chemical interactions takes place between the sample molecules and the particles of the column. They travel according to the relative affinity towards the stationary phase. Because of this, the separation process observed. The mobile phase passes through the pumps, then later on it mixed through the mixture, then later on the solute particles we injected through the injector, then the whole mixture is passed through the column, where the separation process occurs, then later on the separated particles that are detected by the detector sends signals to the system, then later on the whole waste solvent is collected by the waste solvent collector. HPLC unit consists of a solvent delivery system and pump, injector, column oven, detectors, data processing unit. Drug description. The name of the compound is S-naphin malate. It is present in the white powder. The function of the S-naphin malate, it is an antipsychotic drug. The solubility of S-naphin malate, it is highly soluble in water and it is also soluble in dipotassium hydrogen orthophosphate at pH 6.7. The molecular formula of S-naphin malate is C17H16ClNO C4H4O4. The molecular weight is 401.84. The trade name for drug product is Sapharis. The detection of impurities in between the limit of 0.01 to 0.99. The structure of S-naphin malate is shown as an figure. Our aim to find out the impurities of the S-naphin malate. Initially we prepare the mobile phases that is present at the top of the HPLC system. Then later on it is passing through the pumps and reach to the injector. The injector where we should inject the S-naphin malate. Then later on the mixed compound is flow through the column. Based upon the sizes, the separation process occurs and the separated compound is detected by the detectors. In this HPLC system 1090 model, we have the UV detector type. Then later on the detector is sent signal to the data processing unit and that signal is shown as in the form of chromatogram as we shown in the figure. HPLC system for S-naphin malate compound, the impurities is present in between the range of 0.01 to 0.99 limits only. And the GC introduction. Gas chromatography, specifically a gas liquid chromatography, involves a sample being vaporized and injected onto the head of the chromatographic column. The sample is transported through the column by the flow of inert gases mobile phase. The column itself contains a liquid stationary phase which is adsorbed onto the surface of the inert. The sample molecules partition between the gas phase and the liquid phase. Because this partition is critically dependent on the solubility of the sample in the liquid phase. The carrier gas, it acts as a mobile phase. It flows through the pumps and the flow is controlled by the flow controllers. And the later on the injector port is presented. The injector port where we sample we will inject it. Initially, the injector port it maintains certain temperature to vaporize the solute particles which we are injected. Then later on, this mixture is passed through the column. Then the separation process occurs in the column. Then later on, it passes through the detector. Then later on, the detector is passed signal through the recorder. The main principle of GC is it is used to detect the components based on the selective affinity of components towards the absorbed materials. The GC units consist of a solvent delivery system and pump and injector, column oven, detector, data processing unit. Drug description. The name of the drug is esnefin malate. It is present in the white color powder. The esnefin malate is an antipsychotic drug. The solubility of esnefin malate it is highly soluble in water and it is also soluble in dipotassium hydrogen orthophosphate at pH 6.7. The molecular formula of esnefin malate is C17H16Cl NOC4H4O4. The molecular weight is 401.84. The trade name for drug product is Sapharis. Detection of impurities in between the limit of 0.01 to 0.99. Structure of esnefin malate is shown as an figure. Initially, the GC system is continuously flow of the carrier gas. We injected the esnefin malate in the injector port. The injector port is it maintains certain temperature. Because of the temperature, the esnefin malate is converted to the vaporizer form. Later on, this mixture is passed through the column. Then later on, it is separated. The separated column is detected by the flame ionized detector. Then the signal is sent to the data processing unit system and the result is appeared as a chromatogram as shown in the figure.